Alrighty, let's see. Let's go ahead and get started. I'd like to welcome you to the second day of Track C, Data Science Track here at Spark Summit. Thank you very much for coming here to the track. It's my pleasure to introduce Kevin Matter, who is a lecturer at ETH Zurich, also the founder of 4Quant, and here to talk about real-time image processing with Spark. Thank you.
So thank you for that introduction, and I'll start my talk now. So the title is Scaling Up Fast Real-Time Image Processing and Analytics Using Spark. So in terms of an outline for what we're going to do today, it's going to sort of be imaging in 2014, which will cover a little bit of what we saw during Jeremy's keynote talk earlier, and then how we scale imaging up, scaling sort of the post-processing and analysis as well, and then looking at how this applies to real-time imaging and what sort of resources are required to keep up with the flood of information we have, and then sort of beyond. So what do we do next once we've solved these more basic problems with image processing? And so the goal of what we're trying to do is to really have a modern image analysis infrastructure to make these complicated problems and analyses very easy, and to kind of handle the flood of data that comes from the instruments we have now and is capable of handling future instruments which will produce data even faster than we currently get it. So as sort of a look in imaging in 2014, with the constant improvement in detectors and light sources, imaging has gotten much quicker in the recent years. You know, light sheet microscopy, like was talked about in the keynote, can produce images at 500 megabytes a second or more. Synchrotron-based X-ray tomographic microscopy, which I'll explain in the next slide, can make images at over 1,000 frames a second and 8 gigabytes a second of data, which is really an incredible volume to try to deal with. High-speed confocal images can now be over 200 frames a second, which is then 78 megabytes. And there's really many other examples from MRI and other fields of just very high volumes of image data coming in. And the problem with this is that the images are only as good as what you can do with them. And so earlier, if you sort of look at this graph, which is adapted for one that was made for genome biology, because that's another field that's seen some rapid transitions in how fast you can acquire data, you can see that sort of the breakdown between experiment and measurement, so these kind of first two tabs here, the red and the blue, has shifted drastically. And so now you spend very little time with the measurements themselves and then a huge amount of time managing the data and then post-processing it. And so it really changes the way you have to work with these problems, because it takes you 6 months to analyze 3 seconds worth of measurements. And that really is not a very good balance for discovering as many things as you can. So to briefly talk about synchrotron-based X-ray tomographic microscopy. It's a very specialized technique that uses X-ray radiation to look inside samples. And so it's one of the only techniques that can sort of do all of these following because of its ability to penetrate deep into samples. And so as you saw with the zebrafish keynote before, they could look at zebrafish because they were transparent, but they couldn't look at mice because the light doesn't go deep enough. So you can just look at the top layer. And with X-ray microscopy, you can actually go all the way through the sample because the X-rays penetrate. You can get very high spatial resolution, so lower than 1 micrometer with 1.8 millimeter field of view, which means you can see sort of single cells in your sample. You can get above 10 hertz of temporal resolution, so you can get 3D data 10 times a second in your sample, and 8 gigabytes a second of images. And then here are 2 movies. For whatever reason, that one doesn't work. For sort of how tomography itself works, you have the sample here. This is a bone, which is rotated 180 degrees, and you then capture these projection images. And so I'll show it again, where you have these images on the right, which are being sort of the light that goes through the bone, and you get a shadow, like the X-ray you get at a hospital. You can then use filtered back projection and other techniques to reconstruct it into a bunch of slices, so a stack of images that you need to look at, and then do the segmentation and post-processing to get the 3D structures out of these stack of images. So for an idea of kind of the typical large-scale projects that we're trying to work on, one of them is a zebrafish phenotyping project, which is a collaboration with a number of different PIs from different institutes. And that's trying to look at thousands of different samples imaged at a very high resolution where you get 20 to 40 gigavoxels per sample. And so that's a huge number of points to try to deal with. And so when you see, you can get sort of 3D scans through a sample like this, and it's a ton of information to process. And so what you want to be able to do with these data sets is identify single cells, look at the cellular networks and connectivity, classify the cells into different types. So these are, liver cells or muscle cells, and then sort of register it with other types of imaging so you can see where diseases are, or maybe track how fast the animal's growing to understand much better what the genetic background of these animals is doing to the final animal that you see. A similar scale project that we're working on in Switzerland is sort of the brain project, where we're trying to measure sort of a whole mouse brain. And so that's basically 1 cubic centimeter scanned at 1 micron resolution. And so that's 1,000 gigavoxels of data. And so with this, we want to be able to register separate scans together so that we can compare brain structures, particularly as things evolve or change, and then be able to quantify things about the vascular network and other imaging modalities. So in terms of the imaging goals we have, sort of the current state is there's many, many image processing tools available. Some tasks are easy, some are very difficult, and much of the code is sort of highly optimized. So it's written for GPUs so that it's very performant on one machine, but as soon as your GPU runs out of memory, it stops working. And so kind of the issues we have are with this GPU, with RAM, with optimized code, which is very unreadable or untestable because it's written for this one specific application and it's very well sort of compacted for that, but it's difficult to figure out what's going on. Each problem is sort of a little bit different. And I would say the biggest issue we have here is that the scientific questions don't really equal the software packages. And so what you are trying to find from your data is rarely a button you need to click in this program. You need to sort of explore and compare many different things, and that's currently not very difficult in these pre-made tools. And so kind of looking at what we need, or imaging scientists need, is it needs to be able to handle small data sets very quickly so you can get feedback on your results to see what, or in your experiments to see what's going on. It needs to handle sort of larger data sets in a reasonable amount of time so that you can get these analyses done without having a 6-month backup of data. And it needs to scale to enormous sample sizes, so significantly larger than what would ever fit in a single computer's memory. And then, of course, because we're scientists, we don't program very much, and so it needs to be relatively simple, so not much more difficult to use than Python or MATLAB, where you can write one program and have it run on your own computer, on the cluster, on the cloud, and work identically so that you don't need to worry about all these other complicated issues. It sort of needs to be testable, and it would be nice if you could just add computers in to get more computational power. So going back to sort of existing tools, because before you jump to sort of a completely new platform like Spark, you need to be quite sure that there's nothing available now that does what you need. And so usually they have a very friendly GUI, so you can interact with them easily. They're optimized. They scale well up to about a gigavoxel, but they tend to do very poorly above that point. And sort of the negatives, which have been the negatives for many different platforms, is if one machine crashes, everything crashes, and so they're very unstable for large data sets or cluster computing. Yeah, it's tedious to get these programs to do many data sets exactly the same way. Arrays, which are sort of the commonly used structure for 3D data or image data, don't handle multi-channel data very well. And so if you have an array and you want to have a color image, so each channel is a different color, you then need to have 3 arrays. If you then have spectral data, you need to have hundreds of arrays. And so it's very difficult to do analyses with data structured in that format. And implementing new algorithms tends to be very difficult because if you want it to run on the GPU, you have to write specialized code for that. If it runs on the CPU, you have to have something special for that, and you need many libraries, and so it's sort of tedious to use. Yeah. And extracting sort of specific metrics where you're trying to kind of explore your data is typically pretty difficult. Yeah, and checkpoints. So I would say kind of the biggest problem we have with this is that you don't ask the questions you should, you ask the questions you can, which sort of goes back to the scientific questions is not software packages. And so if you have a specific tool like Excel or any other tool, you do analyses that Excel is capable of doing, not the analyses you're actually interested in to understand your samples. And so you need a tool that doesn't limit your imagination when you're trying to explore these data sets. And so most of these tools make it very difficult to kind of play around or explore with what you're doing. Yeah, none of them are very robust or sort of deal with streaming or flooding in data. And 8 gigabytes a second is really a huge amount. And so it's 3 times as many images as Instagram gets a day, at least as of, I think, a couple months ago. But it's a very large amount of data, and so to be using one computer with some off-the-shelf package is probably not the right solution. So to kind of make the connection between this and machine learning, imaging problems or kind of imaging science in general tends to be sort of a machine learning type problem, or there's many similarities between them. And so you kind of have these inputs for your samples, which are the genetic background, sort of the composition you're using to make up your sample, and things like environment. So if you do have a treatment you're doing to these animals that you're looking at, or samples that you're looking at, if you have different temperatures or mechanical testing, that all sort of factors into your input, and then you put it into this imaging box, and you get many of these outputs out. So cell count, shape, distribution, thickness, structural parameters. In the case of other modalities, you get functional imaging, so are these neurons firing or not? And really what you want to do is you want to establish a relationship between these inputs and these outputs. So if you can understand how genetic background affects thickness, you can sort of establish a model for what's going on in your system and how you can tune the parameters in your final animal based on the model you've established. So it's really important for sort of decomposing these complicated systems into simple parts, and you just need to measure lots of data in order to have that. So here's an example from a phenotyping experiment we've did where you have 2,000 bone samples here, you measure them and post-process them. In the same time, you take the genotypes of all these different mice, and then you sort of compare them together to figure out which genotypes are correlated with which structural parameters, like thickness, or the number of channels that are flowing through it, or the number of cells that you have. And so by taking these 2 pieces of information and studying them together, you can establish these correlations. So for the solution, we came up to these problems. It's sort of a Spark imaging layer, and it was developed at sort of ETH Zurich and the Paul Scherrer Institute and 4Quant, and it's a flexible framework for image processing and testing, and so it sort of gives a modular design with these processing plugins that then go into analysis plugins and scale up to cluster or cloud scale computing systems, and give us the ability to do whatever analyses we want, because you can simply combine the tools as you like, and the ability to sort of do parametric assessments of our samples. So you can see how does changing some of the input parameters affect what you finally get out of the system. So in terms of performance on how well sort of Spark or Spark imaging handles our data sets, you can see these are some sort of simple tests that we've done with the number of cores and the voxels per second that these systems could handle, and it scaled very well up and beyond 50 cores. And then this is an analysis of sort of the overhead, so how much of the actual time, computing time was spent doing the computation, and how much was spent serializing data, sending things back and forth. And so we see with a lot of these that it's above 80%. And so that's a very good sign that these tools are working well and will handle our data sets fine. I mean, it's difficult to necessarily say what happens when the number of cores goes from 44 to 1,000, but at least for these size samples, it worked very well. So we do try to extrapolate a little bit to figure out how many nodes we would need to use to sort of keep up with the imaging data flood. And so, you know, with the samples we measure right now, about 1 scan a minute is as fast as we can realistically scan at the moment. And that, I think, falls at, yeah, I mean, so at one of these points. You see these curves and these red lines represent sort of our different goal metrics. And you can see that with our current computing systems, we can handle sort of up to 0.067 scans a minute, which is better than, much better than we've had before, but obviously nowhere close to sort of the future of full 8 gigabytes a second coming in. And we see that even the Milky Way 2 cluster in China isn't quite capable of handling that. And that's assuming we can linearly extrapolate our current performance, which is rather unlikely. But at least we have sort of a potential solution to this problem, whereas before we had no idea how we would go about doing it. So in terms of real time with Spark streaming, and so before, as is very common with sort of scientific approaches, there are many, many different tools involved that are hacked together to get something to work. And it's functional, but if one part breaks, the whole thing falls down, and it's, you know, if you add one node, it won't automatically be integrated into the analysis, and it doesn't keep up with the current data rate. And so using Spark streaming, we can have sort of a 0MQ pipeline feeding the data from the camera into our analysis system, and then we can have a setup like this where we can use as many nodes as we need to keep up with the acquisition rate and the analysis. And, you know, if our data sets get larger, we just add more nodes. And if one node crashes, it continues running just slower than it was before. And so using these kind of tools enable us to keep up, or will enable us to keep up much better with the rates that experiments happen at. So going beyond image processing, so kind of an important choice when you choose an analysis tool is how flexible is this tool for what you're doing in the future. And so the first tests we had were just kind of the image processing and segmentation and analysis. But one of the major things that's done with these data after you've done that are sort of simulations. And so for the example of sort of a finite element analysis, that's actually very closely mapped to what's being done with projects like GraphX. And so you have a graph and you have these connections, between the different nodes, and you measure the forces across these nodes in order to figure out if this point is moving or not. And so with this, we can just immediately move all of our data into GraphX and do these kind of analyses with the same cluster and the same tools without rewriting everything or importing and exporting the data again. Similarly, for other types of simulation, so for cell growth, there's a tool called the cellular POTS model, which is used to model sort of how tumors grow or how cells divide. And this is a similar sort of technique where you can use this GraphX approach to create the network from your imaging data and then study how these networks change with time by applying these relatively simple rules to each node. And so you see that for sort of a standard data set, the number of elements we would have would be around 14 billion, and there'd be 378 billion edges. And so it'd be quite a complicated problem to solve, but we just input it into Spark and GraphX, and it figures out how to do all the really difficult tasks of partitioning the data across different nodes and making sure it's fault tolerant and everything else. Whereas if we try to write a code base ourselves, we spend 90% of the time figuring out what happens at the edges between two partitions and how you synchronize data back and forth, and that's really not problems we're good at solving. So in terms of kind of beyond, as I think some of the presentations yesterday mentioned, they have this BlinkDB, which is coming out of AmpLab, and that's actually even more interesting for us because with a lot of image processing tasks, you usually downsample to make your analysis go faster. But that's, of course, only a kind of approximation of your structure. And you lose lots of information when you do this downsampling. And if you use a tool like BlinkDB, you can get results quicker without making such kind of a brute or a blunt downsampling. You can just have BlinkDB decide which ones are important or which ones can be computed quickly, and you can get your result much faster without resorting to these tools. And so it will give you a much quicker result without losing as much information, or you can control how much information you lose in your setup. And particularly for things like filters on images, if you're just trying to blur your image out, you probably don't need all of the neighbor pixels, you just need a couple of them. And so that would be a result that could be done very quickly without necessarily downsampling. So in terms of kind of a reality check for how all this Spark stuff works, it's not perfect. And I would even say it's not very performant. And so, dedicated, optimized CPU and GPU codes have, at least in our experience, wildly outperformed Spark when run on a single node. And so we won't be able to match the performance of these CPU and GPUs with Spark without having many more computers. But using Spark makes your life much easier. And so it's sort of a trade-off you have to be ready to accept. Particularly with our imaging data, we had lots of issues with serialization overhead, because in order to send these data blocks back and forth, they need to be serialized every time. And it works well until you sort of saturate your network, and then it works very poorly. So when you're sending so much data back and forth constantly across the network, it can be a problem, and it actually starts to affect other people using your system for other tasks. And so that was also less than ideal. But to sort of end on a good note, Scala, Python, and Spark is substantially easier to write and test than any of these CPU or GPU codes. Human time, at least in Switzerland, is 400 times more expensive than AWS time. And so I think that's a very important factor, that every extra minute you spend debugging GPU code, you could have had 6 hours on Amazon's cloud. And mistakes due to sort of poor testing can be really fatal. And so if you aren't sure your code's doing the right thing and you analyze several thousand samples, it's difficult to go back and reanalyze them again. And so with Spark, or with Scala, you can have very simple codes that are very easy to look at and check, where the C++ codes can be a disaster to try to figure out what's going on. And Spark is hardware agnostic, and so you don't need to worry about drivers or vendor lock-in, and to get it running on Amazon or your own cluster is very easy. So Spark is a very flexible solution for these large image processing tasks, and sort of these coarse-grained operations require quite a bit of rethinking, and so all the standard image processing algorithms you've seen don't directly apply to using sort of functional programming-based solutions, but it's sometimes a good thing, because a lot of these old algorithms are very painful to work with. There's, of course, bottlenecks, but I don't think they're game killers. I think they just require a little bit more work with optimizing the Spark code, optimizing usage of CPU and GPU resources when they're available. And I think that in the future, many of these performance issues will be sort of negligible, or hopefully they will be. So in terms of 4Quant, which is the company I mentioned at the beginning, what do they do? And so they sort of do image processing and analysis as a service. And so they'll do measurement analysis and statistical analysis of large image data sets. They'll set up image processing tools on sort of a cloud setup so that you can upload your data and do your analysis there. We develop custom analysis solutions. So if you have a very specific problem, we can combine our tools to come up with the information you're trying to extract from these images. We do consulting on these sort of imaging tasks, and education and training. And so some of our courses are even available online to go look and see or get an idea of how quantitative imaging works and what sort of information you can expect to extract. So for my acknowledgements, I'd like to thank the IT department at the institute I work at, Paul Scher Institute, the rest of the X-ray tomography group, which is called Tomcat, and we're interested in partnerships and further collaborations to make these tools better. So thank you. We haven't really looked at that. That's one of the very new things we're trying to do. We were just seeing, now that we've adopted Spark, what other cool stuff can we do with it? And so maybe I assume actually there's many more optimized tools for doing these problems, but Spark just lets us have one framework where we don't need to switch programming languages and learn a bunch of new APIs every time. Because GraphLab or GraphX, when you're using Spark, is very easy to adopt. So.
There was a presentation yesterday about support for sparse matrices in the MLlib 1.0, and we talked a lot about how always it's going to be better from a storage perspective, and often from a computation perspective, it's going to be a lot more efficient.
Yeah.
Do the optimizations that you guys have incorporated already done incorporate sort of taking advantage of sparsity, or does the data that you work with not lend itself to that?
Well, so I was at that presentation. It was very interesting yesterday. I thought they did a good job. And one of the things he said at the beginning was that you sort of have your sparse data and then your noise on top of it, which then makes it not sparse anymore, which is what we have. And so we have sparse structures ideally, but you need to get the noise out first. And so the images we all acquire are very dense. And so it takes a very long time until we get to sort of this sparse state where we've cleaned out all of the noise and the artifacts in our images and have sort of the underlying structure we're looking at. Once we have that structure, we do store it in a sparse manner. So rather than storing a huge array, we store each point that's available, which makes many of the coding tasks simpler and is much, can be hundreds of times smaller for those data sets. So, yeah.
Is any of the stuff that you're working on open source? And if so, does it work with 2D imagery like satellite stuff? And then I have a follow-up question.
Yeah, so some of it is open source. I mean, we're moving in that direction. It's just we developed lots and lots of tools before that are closed source, and we're not necessarily going to open those up, but a lot of the new things we have are open source. And we even have, so as part of the course I had, and then as part of these slides, which I believe will be made available on the Spark Summit website, we have some sort of basic sort of how do you get started doing imaging tasks in Spark, where you see here, this is how you load an image and save it into this key value format. And then how can you do sort of simple tasks like performing a threshold on this data, looking at a region of interest, doing a box filter. And so what you see here is that a lot of this code, to do a box filter in MATLAB is very easy because you just use the function that's built in, but to write it yourself in C is much more complicated than this. And so Spark makes complicated tasks fairly easy. And when you run this code, it automatically runs on your cluster, your cloud, or whatever you have set up. You don't have to redo anything. So.
The follow-up to that is you said that there's a lot of network overhead and congestion that can become a problem.
Yeah.
Is that because you're sending lots of images around, or are you working on individual pixels and pixel neighborhoods, and you're sending that around the network?
Right. So we're sending.
What's the unit of that you're working with?
So we're sending pixel neighborhoods across. And so with image processing, you always, or in most of the operations you do, it's not really a clean map reduce modality, that you sort of have these neighborhoods you need to be worried about. So when you do a Gaussian filter, you need to look at all the pixels that are surrounding your current pixel. And so there needs to be information that's shared back and forth. And so it's sort of inherent to the problem that a lot of this data will need to be sent back and forth. And Spark, at the moment, with its partitioning, isn't as smart as it possibly could be, because you could save a lot of time if you're able to identify that these partitions are never going to move and these are the only ones that need to be sent back and forth. But right now, a lot of the partitions get sent back and forth that don't necessarily need to, and that creates, of course, extra overhead for us. So.
Do you inference in networks and microprime fields?
Sorry, I couldn't hear you.
Graphical networks or image analysis and microprime fields, do you use them in Spark?
No, we haven't used them at all so far. Sorry.
We get a breakout in the back.
Okay, that's fine. Perfect.
Thank you very much. That's awesome.
Yeah.